Can new genetic information be gained by an organism? A fundamental question in biology is how organisms adapt and change over time. Every evolutionary biologist and creationist alike agree that organisms are not static over time, but have the capacity to change. That change includes the acquisition of new characters not observed in prior generations. But are these new characteristics or physical traits that provide greater fitness in a particular environment the result of the acquisition of new genetic information, or might we say new genetic code? Or as creationists, both young earth and progressive creationists and intelligent design varieties, are these new traits solely the product of simple rearrangements of pre-existing genes and alleles, allowing for new expressions of, in quotes, information that was already there? For young earth creationists in particular, adaptation and thus evolution is ultimately very limited in its extent. In their view of the limited capacity of organisms to change, some new species may form from common ancestries endowed, or they may say pre-programmed, with genetic variation, allowing for diversification into separate pools of variation they call species. But for true changes of one kind of organism over time into a new type or kind of an organism, they might say the ancestor, maybe they might say something like the ancestor of a dog and a cat, with fundamentally new traits, must be based on the gain of, in quotes again, new genetic information, which in their view is impossible since no new information has been added to this world since God's week of created actions. Evolutionary biologists agree that for organisms to truly change significantly over time, such as a gray wolf and a red fox evolving from a common ancestor, and hence a common ancestral genome, those organisms, which are both quite divergent from one another, but also divergent from their common ancestor, must have acquired many new traits, some of which are the result of pre-programmed original information through mutations or acquiring new pieces of genetic code and finding functions for that code through countless mutations over time. Now, this sort of change, which I've just described, and evolution of new characteristics, which is based on new genetic information, that's an anathema to most creation scientists. Let's take a look at that. So let's examine some of the creationist claims, and then we'll begin to take a look at some responses to that. Creationists frequently claim that new information can't be gained by an organism. Here's several quotes. Duplications are the result of duplicating existing genetic information, and mutations alter existing genetic information, whether original or duplicated. Neither of them adds new information. What Georgia Purdom in Answer in Genesis is doing with this quote is she's saying that she's responding to one of the common mechanisms that is proposed for how new genes are created, which is that one gene gets copied, and now there's two copies of that gene, and then one of those genes, because there's a copy and it's duplicated, one of them can gradually change its function over time. And she's trying to claim that that isn't actually adding new information since it was already previous existing information and you're just copying that information. Um, that's yeah, there's a little bit of some semantics going on here, but the main point for what I'm going to talk about is that she's denying that any new information is created. Like this is an example, not in her mind, of new information being created. Second quote: evolution simply lacks the mechanism required to create new information. All right, just a straight up claim that evolutionary biology can't find a mechanism for creating new information. Now, of course, the word information is not defined here, and this is part of the biggest part of the problem is exactly what is that information. For neo-Darwinism to be demonstrated, brand new specified information needs to appear integrated into the original and producing a novel functional structure. This has not been demonstrated. All right, now Brian Thomas of the Institute for Creation Research, he really ups the ante or creates a very specific definition for information or genetic information. You see what he's doing here? He's saying brand new specified information, or I guess specific information, needs to appear. So an organism is there and all of a sudden it adds some brand new information, right? It needs to appear. It has to be integrated into the original genome. So this organism had a genome, and then all of a sudden, new information is added to it. And that new information has to provide a novel functional structure. It can't just be pieces of junk, all right, or just random portions of new code, A, T, C's, and G's that don't have any they don't add any fitness or any advantage. They're not giving an adaptive advantage to that particular organism. He says this has not been demonstrated. This is a very important quote because I'm going to be addressing this specific quote with a very specific example from the scientific literature in just a few minutes. That's the whole reason we're here. But I want to continue on this theme of showing that young Earth creationists deny the ability or mechanism or the existence of new information being formed, and thus not allowing organisms to adapt beyond a certain point. So this is a quote from Kevin Anderson in an article from the Answers in Genesis website. How are new genes made? And in that article, he runs through two or three hypotheses that evolutionary biologists have put forth as to how new genes and new genetic information can be incorporated into organisms, giving them new traits, new adaptations to their environmental circumstances. And for each one of those, he says that this isn't actually new information. Well, either denies that that really can happen or that it doesn't happen enough to be biologically relevant, or he says that it's not actually new information. And again, kind of redefines information in a way that allows him to escape being able to say that it is. So then we get down to the very final quote from this particular paper, claiming that his model or the creationist model of genetic information best understands genomes. Instead, we find the evidence fits within the biblical creation model, where humans, animals, plants were created with a fully functional genome. Since this initial creation, so this is the idea that God has created an initial genome in all the different types of organisms that is fully functional. And I think if you press them further, they'd say it's a very good genome, right? It's a perfect genome. It has everything that that organism could need, and there wasn't anything wrong with it. All right, no negative alleles, perfectly fit for its environment. Since this initial creation, subsequent changes in the genome have introduced many mutations and other alterations to the DNA, right? So that's an acknowledgement. Yes, genomes change over time. Every organism we see, there are changes to the genome, but those changes are mutations. All right, and so those are alterations of the DNA, either at individual bases, so A's and T's and C's and G's are changing one to another, or maybe entire genes are missing or portions of DNA are being moved around, right, within a genome. So they're now the genes are in a different order than they were in the original created organism. Some of these have even provided a specific, although likely limited, this is really important for the creationist argument, adaptive benefit. So this is a begrudging admission that some of these mutations or alterations of the DNA have provided a specific adaptive benefit. Right? So for example, antibiotic resistance in bacteria provides a local adaptive benefit in an environment where that antibiotic resistance would be useful. And so they will say that yes, some mutations could result in some benefits, though ultimately they're going to say that didn't change the bacteria into something else, though, right? They're just going to say that that's a limited local change. Yet these, and here's the kicker at the end, yet these benefits result from degenerative mutations, not the formation of new genes. So he's going to say is there was this original like perfect gene, then there was a mutation. That mutation actually degenerated the original gene, the original sequence, such that it isn't really as good as it was before. However, because the world has changed, like sin has brought decay into the world, and there are environments where, hey, other organisms trying to attack that particular organism, that change allows it to adapt and survive in that situation, which we would say confers a benefit to that organism. But that organism never really needed that benefit in the original creation. And how did they get that benefit? By degenerating the original good creation results in some benefits. I know that's kind of a head spinner there, but this is the way they're looking at it. This is the way they turn any what seems to be a beneficial mutation into something that is not adding something new to an organism, because it's not truly new genes. It's not like a whole new gene. It's just adapting a previous gene, and usually it's not even as good as the original gene, but it may have some benefit in local circumstances that you could call a benefit, but don't call it a universal benefit, because that would suggest that the organism could have gotten better compared to its original created form. So now I think this kind of establishes the creationist mindset with respect to an organism's genome, what it's capable of, and what its limitations are. Oh, one more slide, one more illustration of this. Here's a screenshot from a talk given at the Creation Museum by Answers in Genesis, and here they have a slide on the screen in front of the speaker about mutations. What are mutations? And so we see here all mutations, even beneficial ones, and of course putting beneficial in quotes, as I was just saying, right? You can't really just completely acknowledge they're beneficial. You have to slide like locally beneficial, like they could be called beneficial by some in some circumstances, are a loss of variation of pre-existing genetic traits. So, and then the easy example here is to say, oh, look, this owl here has a lot of genetic variation, can make a variety of different coat colors. And then if they have a mutation to that gene where they lose the ability to make melanin, they then become albino. All right, so that's just changing the gene through a mutation, and it's a loss of function. But that loss of function results in a white animal. Now the speaker goes on to say, now, what if this owl was living in the north where there was a lot of snow cover all the time, and so the white color actually confers something of an advantage to that organism? If that advantage allows it to survive maybe better than the brown-coated owl, which lives maybe farther south, you could say that that mutation confers a beneficial effect, right? But you could also acknowledge at the same time that's actually breaking what was previously a functional gene into a non-functional gene, and you can't say that's adding new information. You can call it adding a new trait, all right? It's a new trait, but it's not actually adding new information. It's simply breaking information that's there before. Again, it's hard to talk about this language of information without a definition. I would love to give you a definition, but young Earth creationists, and really the word information itself is a loaded term, even in many secular disciplines. So we're just trying to get the ideas of what creationists say about information before I then tell you what I'm going to do here. And then she also throws in here, most mutations have neutral or negative effects, right? Just the reminder that most mutations aren't good, and so therefore she's giving you the perception, at least, that mutations basically tear down organisms, and she's already given you an example of how this mutation causes these owls to lose their coat color, which in certain circumstances may be beneficial, but in the long run, it's certainly not going to change. And this is the whole emphasis of all this. It's not going to change an owl into something that's not an owl, right? It's not going to change an owl even into something that's a different kind of owl. Um, it's simply a variant, and it's even a loss of variation. It's sort of the lowest level of variation. It's a broken version of a gene. Right, that all sounds quite compelling. And as speakers at Answers in Genesis, and I recently went to a talk at the Ark Encounter in which they talked about mutations. It's very compelling to the audience. It makes a lot of sense that there is information and most mutations are bad. I don't disagree with this last statement. Most mutations are neutral or negative. That is an accurate statement, but it is an accurate statement that presents the perception that somehow, because there's so few beneficial mutations, that no organism could possibly create new information or truly adapt new traits based solely on a few beneficial mutations. All right, so now comes the question. Is this true? Well, let's find out. And my goal here is not to be comprehensive in my response to these statements about mutations and lack of new genetic information, but to simply provide one single example from the literature um, to give you an idea of to give you some insight into the complexity of this world of information, and I think to provide you an example directly of an organism that has gained new genetic information over time. So my objective, demonstrate that some organisms have obtained a novel function via the acquisition of new genetic material, not possessed by other members of the same family or even kingdom of organisms. So I'm going to try to show you that there's an organism out there that has gained a new, not just new DNA, the raw resource of genetic information, but has taken that resource and developed it or used it for a new function that its relatives that don't have that particular piece of DNA can't perform. And so this speaks directly to Brian Thomas's quote, in which he claims that this has not been demonstrated, right? That an organism can suddenly have new genetic information that wasn't there before. And not only is there new genetic information, but it's actually useful for a function and provides an adaptive value to an organism that didn't exist before that. So to achieve this objective, what do I have to do? Another way of putting this, what do I have to do to convince you of this, right? What do I think I'm going to need to provide you with? First, I'm going to have to provide you with some knowledge about prokaryotic and eukaryotic genomes and how they regulate and read their genes. In other words, I'm going to have to teach you some biology. Specifically, I have to teach you some genomics and, well, genetics and gene regulation. All right, so I gotta lay the baseline of the biological knowledge you would need to know to understand my example. Then I'll need to show that the genes are novel to a group of species and that they utilize them to produce a function beneficial in their environment that their relatives can't perform. So I have to show that there are very closely related species of a particular organism that lack this function and lack the code, the genetic information to perform that function. But then there are other related species that have this code and they can perform a function that their friends and neighbors can't, right? And then I will need to explain some of the mechanisms about how these genes not only were gained, so how did they get that new information, right? Where did it come from and how did they get it? And then how did they use it? How did they actually take that new information and use it as a new phenotype, as a new trait for that organism, giving them a truly novel characteristic that other members of their particular group of type of organisms can't perform. All right, this is a big challenge. So now, you ready? All right, warning, super nerdy molecular genetic science talk coming up. Right, we're gonna get pretty detailed. I'm gonna try to explain everything in every step. I may go into far more detail than you need. I may go far more detail than you can handle. But I'm going to start with some simple ideas and hopefully build your knowledge level up so that you'll be able to, I'll say it this way, so that you can appreciate the amazing example that I'm going to provide. But also, I want you to appreciate the amount of work and the amount of skill and the amount of research that's been done in this case by a very large research group to do this. Okay, so if you're ready to take on the challenge, you're ready to get a little nerdy with science, we're ready to go. We're going to start with prokaryotes versus eukaryotes. Right? This is a fundamental distinction between two basic types of organisms, right? We could split, if we took all living things on the face of the earth and we said, I just need to divide them into some major types of organisms. We could divide them into two major types. I could put them into two different pools. baskets, right? Prokaryotes and eukaryotes. And the simplest way to think about it is prokaryotes are the bacteria. There's actually two kinds of bacteria, two major groups of bacteria called the eubacteria and the archaebacteria, but well, they're just lumping them together, bacteria. And then you have something called the eukaryotes, and that consists of the protist, single-celled organisms that aren't bacteria. Then you have your plants, your fungi, and your animals. Those are all prokaryotes. Now, what makes a prokaryote different than a eukaryote? Well, they both have the word karyon in them, which means nucleus, all right? And pro in Latin is before. We scientists like to use that ancient language Latin in our terms. It's really helpful to know Latin, right? Because then you just know what the definitions of a lot of scientific terms are that way. And eu means true. And so down here we have a bacteria, a prokaryote, and that prokaryote is very simple. Bacteria are very simple. They have a plasma membrane, the outer membrane of the cell, and inside it's just a fluid sac. And inside this fluid sac, you'll find the chromosome of the organism, which contains the DNA. And it's just one long, big, long circular piece of DNA all sort of bunched up inside this cell. And there aren't any other internal organelles, internal large structures. Whereas a eukaryote, such as yourself, you have a nucleus, right? You take your DNA, right, your chromosomes, your genetic information, or we'll call it, the genetic information, the genetic code, and you place it inside the nucleus. And the nucleus has its own plasma membrane around. You can think of it as a cell inside of a cell. And then there's a bunch of other types of cells inside your cell as well. Mitochondria, plants have chloroplast, and this is the endoplasmic reticulum and so forth. So there's all these other structures that are in there, making the eukaryote a much larger, far more complicated cell than a prokaryote is. And so, in fact, the sizes here are not anywhere close to being relative. This bacteria should be more like this size down here. I'm probably even drawing that too large, right? You can fit usually tens of thousands, maybe even 100,000 bacteria inside of a single eukaryotic cell. So that's prokaryotes versus eukaryotes. Now, why am I even talking about these very large distinctions between organisms? Because the example that I'm going to give you of new information in an organism involves the transfer of information from a prokaryotic organism, moving it to a eukaryotic organism, inserting it into its nucleus, giving this eukaryotic organism a new function, a new property that it didn't have before. That's it in a nutshell. That's going to take me a while to explain how that might occur and what the significance of that particular transfer event might be. So prokaryotes and eukaryotes. So now let's look quickly at the genome of a prokaryote, and the prokaryote I've chosen to show you is E. coli. E. coli. E. coli is that bacteria that you have hundreds of millions of inside of your intestines that are helping with your digestive system. And it's one of the most studied organisms on Earth. And this is a complicated looking graph, but all I want to show you is the genome of this organism is 4,938,875 base pairs, at least for this particular strain of E. coli. There's a lot of different E. coli strains that have different sizes. Oh, that should tell you right away. The fact they have different sizes mean that different E. coli have different size genes, or they have inserted pieces of DNA or pieces of DNA that have been lost from their genome, and that affects their capacities, all right, their functions of different E. coli strains, which is why some E. coli strains can eat the flesh off your face, while as other ones can live quite comfortably in your stomach without hurting you. Anyway, I just want to show you this outside portion shows where the genes are. All these little bars here, the blue and the red bars around the outside just represent all the different genes in this particular organism. And the critical feature of this prokaryote is their genes are usually in groups, and those clusters or groups are called operons. So we're going to have to talk about operons for a moment in order for us to understand the significance of this transfer event that I'm going to relay to you. One other thing I would like to point out about the genome of E. coli is that there's very little space between the genes. So as you're going around this, there aren't large segments, and I know you don't necessarily understand what all these little pieces are, but I'll just tell you there aren't large segments of code between genes, which we call spacers or non-coding sequence between them. So this is a very compact genome in which the organism is literally almost using its entire code. You can think of it as like the genome is like a hard drive, right? It's the space where you can put your programs. And these are the programs that run a cell. And so for E. coli, it's as if they have a hard drive in which they are using 95% of that hard drive for programs, for genes. Whereas your hard drive is absolutely massive, right? Your hard drive is for each copy of your genome is like 3.3 billion ATCs and Gs instead of 4.9 million. And 3.3 billion, but you're only using a very small percentage of that, just a couple percent for the codes that make up most of the programming of your cells. And you have a lot of other stuff out there, which we're not going to talk about today. But that's a big difference between most prokaryotes, which are very compact in terms of their codes and their hard drives, and eukaryotes, which generally have lots of extra space. It's like having a giant hard drive, on a brand new computer you bought, and you have an operating system plus a couple programs. But in a sense, you have a lot more room for additional programs, right? And that's really important to remember as well. Not necessarily for this talk, but I think it's a good thing to remember is that um, eukaryotic organisms have an abundance of an amount of hard drive space that they aren't really using for specific functions. And so it becomes, in a way, highly adaptable for deriving new things that they can do. They can play around with that stuff because it's not essential. They're not essential components of a genome. But I digress. So back to the bacteria. We have to talk about one portion of a bacterial genome, and that is some of their genes and how those genes are regulated. Now, gene regulation is about how do you find a program that you need to run in your genome? How do you turn it on? So you got to find it in your genome, just like you have to find a program on your hard drive. Then you have to turn it on. You have to express it. That's like copying your code out of your hard drive, and then you have to translate it into the parts that you're going to make. In other words, what is this program saying to make? Well, that's what your CPU does, right? It reads this code from your programs and it makes it into stuff. Well, what you do and what these bacteria do is they have a series of genes. So here are these, in this case, structural genes. A, B, C, and D are 4 different genes, all very compact right next to each other. And upstream from that, just meaning before these genes, there is a portion of the code, something called the promoter and the operator. This is where large proteins that are called DNA binding proteins can bind to the DNA and stimulate the copying of those particular genes. So let's say that there is lactose in the environment of a bacteria, and that's a specific type of sugar. And it's not normally around. So normally what happens is you don't have these genes on. You're not expressing them. You're not making the products of them. And what are the products of these genes? In this particular example, we could say that the products of those genes are protein A, B, C, and D, and those proteins are enzymes. And those enzymes go about helping you to break down lactose. So if you had those 4 enzymes active inside your cell, you could take lactose from the environment, break it into little pieces, and get energy from it. That would be a very valuable thing for the bacteria to do. Now, if lactose is around, you have to, there has to be something that triggers the bacteria to say, hey, I need to, use these genes. Well, I got to go find those genes. So that's what the promoter is. You're going to promote, like copying these genes. The operator also is something you can bind to stimulate, or you can block the progression of it. But let's just say you're stimulating the what's called transcription. Transcription is copying this code. And so if all these things up here, and there's also something called regulator genes, which also make proteins, which then can bind to things and so forth. So there's all this other information that goes along with genes themselves that's part of the genetic code that helps you operate and understand your genome. And so let's say so lactose is around. Something about the presence of lactose stimulates you to bind these proteins, which then turn on the transcription process, which reads your DNA into a long strand of what's called RNA. And RNA is a UCG UUA. It's the same, basically about the same as DNA, except you have U's instead of T's. So it's a form of nucleic acid, and it's a string. It's a string which is a code itself, the messenger RNA. That messenger then can be immediately read by the bacteria by its code reading um, machine called a ribosome, and it will read it into amino acids which make up proteins. And so the sequence for code A becomes messenger RNA code A. It gets read into a protein, which is protein A, and it has a particular function. Maybe it's like, hey, I attach to lactose and I break it into two pieces. And then after I've done that, protein B, which the sequence for that was here, and you read it into protein T. Now protein B takes lactose, the two pieces, and breaks it down a little bit more. And then it starts releasing energy, and protein C and protein D both break it down even more. And eventually what you do is you have this series of metabolic reactions resulting in the breakdown of uh, that molecule and the release of energy which the bacteria can use to live on. So this is a very useful thing. And the idea of an operon is that all of these genes work in conjunction with each other. In other words, when you turn this on, when you promote it, you promote the reading of all four genes together at the same time. And you see how that's useful? Because it's like saying you have an executable file, right? When you click on your computer on like I want to open this program, you click on the executable file and it goes and it finds a bunch of different files and it opens all those files at one time and says, okay, you need all these files in order to do this function. That's what the bacteria is doing, saying like you need all these genes because you need to make all these proteins so that you can break down lactose. And it's nice because you make them all about the same amount because you're making them all at one time. So this is very efficient. And then when lactose is not there anymore, you simply turn this off. And so there's a way to unpromote it, right? Right? You can stop this promotion, which then stops you from reading the genes anymore. You stop making the proteins, in which case you stop breaking down lactose, which is good because you don't want to waste all that energy making proteins if they don't have anything to do. If there's no lactose around, don't make them. Okay, that's a long description of an operon, but I want to give you the idea that this is how prokaryote organisms generally are reading programs and running and operating their cells. Now we have to talk about the difference between prokaryotic organisms, bacteria, and eukaryotic organisms. Now, they're very similar in many ways, but they have differences in the way that they regulate their genes. How do they turn their genes on? How do they turn them off? Um, and so one of the differences is how they promote or read or stimulate their RNAs to be transcribed or their DNAs to be transcribed. So let's just look at this prokaryotic promoter site. And so 10 base pairs, that's what the minus 10 is. Minus 10 base pairs means 10 base pairs in front of the beginning of the gene. So this blue thing is a gene. That's what's going to be read. That's where the code actually is that makes a protein, which is going to have some function inside of a cell. If you go down 10 base pairs, what you'll find is for most bacterial genes and operons, you'll have what's called the Pribno box, which is TATAAT. You'll find this particular code somewhere upstream. Now, why is it that particular code and why is it right there? That particular sequence is recognized by a protein. So there's a protein that the bacteria can make that can recognize this particular sequence in the DNA, even in the double helix when it's wound up. It can recognize TATAAT, and if it sees that, It will help the RNA polymerase, which is the thing that's going to open up the DNA and begin to copy the RNA, to signal to it, hey, we're going to, this is where you can get started. There's also another sequence 35 base pairs downstream, TTGACA, which has some slight variations in different bacteria, but this is what's called the consensus sequence. At 35, and that's also a place where proteins bind. And the binding of these different proteins will then help stimulate or initiate the beginning of reading code. So this is the form of gene regulation, a part of regulation. All right, so that's prokaryotes. Now let's compare that to eukaryotes. So down here we have eukaryotic promoter site. Here again we have a gene. All right, this is where the code is, the sequence of a particular gene, and that gene might be a very similar gene to what we have in the eukaryotes, prokaryotes. There are genes that are in common that eukaryotes and prokaryotes have. If we go 25 base pairs downstream, we'll find a sequence called the TATA box, which is TATAAA. You see, it's not that much different than the prokaryote, but it's typically TATAAA, called the TATA box or the Hodgeness box. And this is also what's called a DNA binding site, where proteins can bind to this particular position, which then helps the RNA polymerase and other proteins that are regulator proteins to bind to this area. And then maybe 75 base pairs downstream, but not always present in every eukaryote, is something called the CAT box. And that's another place where a protein can bind. And sometimes that protein can actually inhibit regulation, or it can stimulate it. So there's different functions. But so just two things I want you to notice here. The sequences are different between prokaryotes and eukaryotes. Not much different, but different, which also means that the proteins bound to them also recognize different sequences from prokaryotes and eukaryotes. And also the distances from where this occurs. The distance is important because it turns out there's other large proteins that have to glom onto this in order to start the transcription. And the transcription machine is different in a prokaryote than a eukaryote. They consist of different proteins of different sizes, and they really need more space. It's a larger mechanism. You can think of it as it's a larger machine in a eukaryote, capable of recognizing a lot more controlling mechanisms, because the machine has other places that things can be attached to it to either speed it up or slow it down or inhibit it or stimulate it. Very complicated stuff. But the point is that they require a little more room for all that to occur. So the identification of this promoter site is farther upstream than the negative 25. Okay, so there's differences between eukaryotes and prokaryotes in how they're regulating or turning their genes on. Now, okay, with that background, prokaryotes and eukaryotes being kind of fundamentally different types of organisms, and they operate their codes different. Kind of think of it as Macs and PCs. They have different operating systems. Yes, there are many common features to them, which is why they can recognize some of the same programs, right? Some of the same files. But they're not perfect matches either. You can't just expect your Mac to be able to read anything made on a PC. It doesn't completely understand the same language. That's very similar to a prokaryote versus a eukaryote. So now we come to the paper that I want to talk about, right? Now we're going to delve into the real details. So I want to talk about this paper, Eukaryotic Acquisition of a Bacterial Operon. Now, I've already given you all these words, right? I've said a bacterial operon, which is what we've been talking about, is a group of genes all copied together and regulated by a prokaryotic regulation set of sequences. So that's a bacterial operon. But the first part of this title says eukaryotic acquisition of a bacterial operon, meaning there is a eukaryotic organism out there that has acquired or obtained a bacterial operon. So they've taken or accidentally been given an operon, and an operon is, you can think of it as a program, right? The operon is a set of genes that all work together to produce a particular function for an organism. So if you're going to procure an operon, it means you're procuring a function for yourself. So eukaryotic acquisition of bacterial operon. This was published in the journal Cell, right down here, in 2019. All right, so this is not very old. It's actually written after one of the quotes I gave you by the creationists. But nonetheless, he's not off the hook for he's off the hook for not knowing about this one, but there are other examples he should have known about before he wrote his statement about this is unknown. There's a lot of authors to this paper, and I only want to say, and I only want to point that out, and they come from a number of different locations, just to say that this is complicated work. The results are quite simple, but their observation of something, I'm going to show you is they observed something as they were researching something else. This is the way science often works, right? They're working on a group of people, a smaller group of people, working on a particular group. It's going to be fungi. And while they're working on those fungi, trying to understand a bunch of other stuff about metabolism of fungi, they stumbled onto, this discovery of something that they found in fungi that they didn't know could exist in fungi. And then they wondered, where in the world did this come from? And so they had a hypothesis, and they needed to demonstrate, they needed to test that hypothesis. And so they actually had to add more people to their group because they had to find a bunch of different expertise, and they went through a lot of different testing of various hypotheses. In order to try to prove their particular hypothesis about what they think has happened here and why these fungi have this capacity to perform this function that was very surprising. And so this is the efforts of a lot of people collecting a lot of different forms of data. And I'm not going to cover all the different forms of data. I'm just going to give you the relevant forms of data they collected. But each one of them is kind of like a project within a project, but it's all put together in this one paper. So I'm going to do this I'm going to do this in kind of I'll call it an iterative way. I'm going to give you the big summary. Then I'm going to give you the summary again, but with more detail. Then we'll go down and talk about the summary. We'll talk about each of the elements of that summary or that abstract and show you their evidence, right, for the things that they're saying there. So we'll start over here with the graphical abstract. All right. I love these papers that now require authors, and I've had to do this for several of my papers, create a graphical abstract, something that visually tries to communicate the basic message of your paper. So let's look at this graphical abstract. The basic message here is there's a bacteria. You're saying there was a bacteria. That bacteria experienced or allowed, we'll have to talk about that language a little bit later. But what happened, or at least what they're showing happened, is a portion of its genome. And so each one of these little blue boxes here, each one of these little blue boxes is a gene. And these genes are all connected together on one portion of the chromosome of this bacteria. And they're colored blue to indicate they come from the bacteria. This little chunk of the bacteria ends up being about 16,000 base pairs long. Was horizontally transferred, or what they're going to call a horizontal operon transfer, a HOT, was transferred to this yeast. And inside this yeast, the genes from this bacteria found a function inside the yeast such that this yeast now has a function that it didn't have before this particular transfer. Right? So this is where I'm going to get this idea that there's an organism out there that acquired new genetic information that didn't exist in that organism before. And not only did they acquire new genetic information, but they put it to use and found a function for it, and they're using it, and it makes them more fit for their environment. All right, so they have a characteristic, an adaptation that is novel and new to that yeast compared to all other yeast. That is explained a little bit more in the additional graphical abstract. They're saying, okay, look, there was this bacteria. That bacteria transferred because yeast lived at yeast have existed at the same time that these bacteria exist. That bacteria then, there was a transfer event, and I'm going to have to talk about that transfer event. Like, why do they think that could happen, and how could it happen? I have to give you some plausible mechanism for how that could happen to make this believable. We'll get to that. That transfer occurred at some point, and after the transfer, some of those fungi, all right, still have that. Some fungi that exist today, all right, live today, have this bacterial segment in them which is performing a function. That function, which I'll tell you now, is making it's a series of genes that make enzymes that are part of a metabolic pathway that the end result is a protein, or I'll just call it a biomolecule called enterobactin. And enterobactin is very efficient at grabbing iron from the environment and being able to bring it into the organism. Bacteria make enterobactin, at least some groups of bacteria make enterobactin. They're proposing that the one that gave it to this fungi was one of those. And they're very good at grabbing this iron from the environment in this fashion. But to this day, at least till this moment in time when these researchers revealed this, no eukaryote and no fungi especially, are known to be able to make this particular biomolecule. Right? So they're not known to have this function of being able to procure or scavenge iron from their environment through this mechanism of making enterobactin. Now, what they also are going to say and discover is that it wasn't just one fungus, one species of fungus or yeast, but they found multiple different species of yeast that all have this capacity. They all can make this enterobactin, and upon looking closely, they found out they all have these genes from what look like to be from a bacteria. But interestingly, the different fungi also are not exactly identical to each other. Some of the fungi have additional genes that have been added to the genome. In this case, they've not actually been added. They've been moved from another place in the genome over into this portion of the genome. And these genes become part of the process of scavenging and making the metabolic pathway more efficient. Now, so fungi, some fungi aren't quite as efficient because they don't have those extra genes, and then some fungi have an extra gene, and some have two extra genes or three extra genes inside of this group. One of these fungi has split the genes that from the bacteria into two different groups in two different places on the chromosome. So the bottom line here is they're similar to each other. They're all amazing in the fact that they have a new feature that no other fungi or no other yeast has. But each individual yeast species has its own unique combinations, giving them their own unique properties and abilities and different skill sets in terms of scavenging iron from their environment. And then, of course, the graph down here is trying to, where's my, yeah, the graph down here is trying to say, hey, there's thousands of other species of fungi that, hey, nope, they can't do it. We've seen no evidence that they have these genes and they cannot make enterobactin. They cannot scavenge iron in this way. Yeast can get iron from their environment, but not like this. That's the graphical abstract. So that's giving you like the big picture. But to convince you this is what has happened, we're going to need to look at some more data, right? We're going to need to dig into this a little bit farther. Let me read some of the summary, the abstract. So now I'm going to give you the written version of the graphical abstract. Operons are a hallmark of bacterial genomes. Well, that's why I just spent some time talking about bacterial genomes and operons, where they allow concerted expression of functionally related genes as a single polycistronic transcript. I didn't use that fancy term before, but copying all your genes all together at one time as part of an operon is how you make a polycistronic transcript. It just means poly means many, so you make many transcripts at the same time. They are rare in eukaryotes. I hadn't said that yet, right? This sort of way of regulating genes and creating entire metabolic pathway sets of proteins or enzymes is rare in eukaryotes. It's very rare in you. All right. You and I and mammals, we read all of our genes separately. Every single gene has its own control. We all have a separate promoter. We individually have to go through and turn each gene on separately. And you might think, well, that doesn't sound all that efficient. All right, because you just made this big deal about how bacteria are all super efficient because they read all their genes together and they can make one program at one time. And now you're telling me human beings have to like do it all separately. The thing is, human beings are far more complicated and we have different types of cells. And sometimes we use our genes for multiple purposes. And so we can't have them tied down to, I'm only going to turn them on when I'm turning these other 5 genes on to do this one thing, because we often do different things. In one cell, we might have a gene on and we express it, but we don't need these other 3 genes. Whereas in another cell, we might need those other genes on at the same time. And so it's just better for us to have individual control, because individual control means far more complexity. So that's the price we pay for complexity, is that we can't keep things simple like these bacteria. All right, back to the summary. Yeah, I can't find, there it is. Each gene, I was just saying this, where each gene usually drives expression of its own independent messenger RNAs. Here we report the horizontal operon transfer of a seridophore biosynthesis pathway, that's the fancy name for making enterobactin, from relatives of E. coli into a group of budding yeast. So they're saying an entire operon, a group of genes that's a program, is moved into a eukaryote. But I'm telling you that eukaryotes don't usually have operons in them and they don't function that way. So how can you move something that functions differently in a prokaryote and has different promoters and different regulators into a wholly different type of organism and get it to function there? Seems unlikely, but stick around. We further show that the collinearly arranged secondary metabolism genes are expressed. Collinearly means these genes are all in an order. They're in a line, and they're all expressed at the same time. They're going to show that. I'll show you that figure in which they show that these genes are being all turned on and transcribed all together, not piecemeal and separately. And so they are expressed. Expressed is just a way of saying they actually make proteins. They're not just a piece of a genetic information that's sitting in a genome, but it can't do anything. No, they're actually turned on and actually made into a product. And not only that, but I'm still in trouble with this, exhibit eukaryotic transcriptional features, and they enable the sequestration and uptake of iron. In other words, they're actually going to prove that the fungi make enterobactin. So the products that they make are a successful metabolic pathway. It makes enterobactin, and not only does this yeast make enterobactin, but they go the extra step and they show that these yeast actually grab iron from their environment, are able to sequester it, pull it in, and keep it inside of the cells in a low iron environment in which they live, which, as you'll see, the types of yeast we're talking about, they live in low iron environments, and having this ability to get iron from their environment and sequester it like a bacteria is a very useful function to these particular to these particular fungi. Okay, I'm not going to read the rest of this. This has to do with a whole bunch of details about how they have changed their gene regulation in order to adapt to being in a eukaryote. That's all important, and I will just touch upon that later. I want to stick with the core ideas. So first thing we have to do, let's start at the beginning. Let's start with, first we have to show, why in the world do they think that these genes that they found inside of fungi are not actually fungal genes, but they are bacterial genes, and therefore must have been gained from a bacteria. So first is a figure 1 from their paper. They were looking at this group of scientists are interested in metabolism, and they're exploring all the different ways that fungi manipulate their environment and are able to uptake energy, different forms of energy, and other types of minerals. right? And as they're doing that, they're looking at iron, because iron's a really important mineral in environments. And they also knew that some fungi live in iron-poor environments. So they wondered, like, how do they get their iron? Now, all these green boxes represent fungi. Oh, I should point. Okay, let's cover a couple things here first. Saccharomyces, that's a fungus. All of these are fungi. The Psidiomycetes, those are your mushroom fungi, your typical mushroom fungi in your yard. And then there's a whole bunch of, most of these fungi up here are basically your yeast type fungi. There's a lot of different kinds of yeast. And then there is one particular group we're going to talk about. I can't really pronounce these. Starmarella and Wickerhamiella, two different genera of yeast. And they said, look, there's all these different forms. There's ceritophore import, there's low affinity iron uptake, there's vacuolar iron storage. They're looking at these different ways that they deal with iron, and these little plus marks say that yes, they can do it. That, this fungi can do this. All these fungi can do this. In some cases, only 10 out of 17 of the ones they looked at. So you see, they looked at 17 species and actually tested them. And only 10 of them could do this particular type of iron, sequestration and so forth. But all right, so the idea is that fungi have a variety of different ways. There's multiple ways to skin a cat, and that's true for lots of different functions of organisms. We're not just dependent on one thing to obtain minerals, nutrients, and food. But over here, there's a certain kind of biosynthesis called siderophore biosynthesis. And there are some fungi that can do a particular type of siderophore biosynthesis that makes that is able to secure some iron in this fashion. But this is where they found something new. All right, and these are undoubtedly fungal genes. In other words, saying all these fungi, of which there's actually thousands and thousands of these Basidiomycetes, they're only tested 21. They have the capacity to do some form of siderophore biosynthesis, but this form over here is considered bacterial form, which means they have a different set of genes and a different order. They make different enzymes, and they produce this particular chemical product as the result of their metabolic pathway called enterobactin. All right, it's got the word bactin in it, right? Bacteria in it. And they found out they've got this peculiar chemical, right, which is procuring iron, or looks like it is. But it's only found in these two genera. And in fact, it's only found in 12 out of 18 of those species. And it's not been observed ever in any other fungi, and it's not known in any other groups of organisms, any other eukaryotes. So it's like, well, where did they get this? Like, how are they able to do it? Because bacteria have this operon. They have this whole genetic pathway, right, for being able to do this. Fungi don't have that. Out of the many, many, many diverse groups of fungi, they don't have it. But then there is just these two yeast, which are very similar to other yeasts, but these two can do it. So now the first thought might be, hey, I'll bet maybe there's some bacteria, and I haven't told you where these live yet. I'm saving some surprises, right? Maybe there's some bacteria that live with the yeast or inside the yeast, because there are bacteria that actually live inside of other cells. Maybe there are bacteria that live inside these yeast that can do this kind of iron uptake, and they're kind of like doing it for the yeast, right? It's a mutualistic relationship, and they're doing it for the yeast. So how would you show that that's not what's happening? What they did was they went in and they sequenced a bunch of genomes. They extracted the DNA from these organisms and sequenced the entire genome of the organism. And when they do that, they would have sequenced any genome of bacteria that was there too, which would be easily identifiable as bacteria. But when they sequence the genome of the fungus, they're reading down the fungus. And as they read down the fungus, let me get my pen out here. As they're reading down the fungus, what they find is that they're reading down its fungi. This is like all other fungi. And then all of a sudden, there's a little segment of the genome on one chromosome of the fungi. So it's definitely part of the fungal genome that is bacteria. This is all bacterial seed. This is a sequence that is just like other bacteria, but not like any other fungus. And then it goes right back to being a fungus. See, millions and millions of base pairs, 16,000 base pairs that are bacteria, and now all of a sudden, a bunch of fungi again. So this just sticks out as like, where did this piece of DNA come from? Looks like a bacteria, but it's in a fungus. Um, okay. Quickly, the biochemical pathway. It's because we're going to talk about some of these genes, not in any detail. You don't have to memorize the names or anything like that. There's not going to be a quiz on this. So here we have int C, int B, int A, and then there's a variety of other genes right here. You notice this int B right here is the same as this one right here. That's because, and this is, I think this is good to know. There are enzymes that can perform multiple functions or functions that are very, very similar, and so they're responsible for multiple biochemical pathways. And this is why our genomes are a little bit mix and matchy, right? This is why when one lets us down, another one can kind of fill in a little bit because they're not all just do their job perfectly and only do one thing. They're often sloppy. I tell my class this all the time. I mean, biochemistry is actually quite sloppy. Most chemicals have a job or a thing that they mostly do, but they also kind of do some other things too on the side. So anyway, each one of these enzymes that's responsible for doing the trans changing this chemical into this one over here, this enzyme is a protein. And where do proteins come from? Proteins are encoded by genes. So there must be a gene. If you find a protein inside of an organism that's doing some job, there must be a gene somewhere, right? A piece of DNA code that is telling it how to make that protein. So there's a code for int C, there's a code for int B, there's a code for int A, int D, int F, right? And int E. And so all of those genes have to exist to make all these proteins in order to do this whole pathway to end up with this molecule, which is the thing that actually does the function that we're going to notice. Let's start with some of the evidence that they have that the genes that they found in yeast are not native to yeast, but probably transferred from bacteria into yeast. So here they did, they compared, so they took some sequence of this bacterial sequence, right? So they took some of the sequence of some of these genes, the int C, int B, and so forth. They took the sequence from the yeast, and then they compared that to the sequences of a whole bunch of different organisms, right? There's a way in which you can just take those sequences and you can do what's called blast it against all the other sequences we know about, across the entire world of organisms. And what they discovered is that all of the yeast sequences they gave them, well, all the sequences they found inside of the yeast genome that made these proteins, all of them were very similar to one another. In other words, the yeast that they compared, because they actually sequenced multiple yeast species, all of them were all very similar to each other. So we'd say they're all related to each other. However, they were, as a group, related to these other groups of things. And down here, here's Escherichia, that's E. coli. And these are other organisms that are related to E. coli. All of these ones that are in red are the ones that have essentially the same sort of genes and similar sequences to the sequences that are in the yeast that they looked at. All right, so how should I put this to simplify the message here? The point would be this green line represents yeast sequence on a chromosome. And then there is this section of red. That section of red is almost identical to a bunch of other bacteria. If you were to take the sequence from this portion of the yeast genome and then compare it to all the sequences out there, bacteria, humans, all the works, you would undoubtedly find that this yeast sequence would just be like other yeast. Because the yeast is, very similar to other yeast cells, other species that are similar to it. So this is what, this was both surprising, but they had already, once they noticed that there was this weird ability that these have that no other related yeast had, they derived the hypothesis, well, maybe this yeast actually gained this function by getting this operon from a bacteria. All right. And so somehow this sequence from a bacteria got inserted into the yeast. And somehow this sequence got inserted after it got inserted into the yeast figured out how to use it, even though it's a bacterial derived sequence, which I've just been showing you, has differences in terms of how they regulate those particular sequences. But the first step is to show first, this sequence actually is related more closely to bacteria than it is to fungi, and it certainly is. All right, I mean, there's no doubt that these sequences are essentially bacterial sequences, but they're found in a yeast. So that's your first piece of evidence. It's considered a very strong piece of evidence that this piece of sequence is not original to the yeast, but is actually original to bacteria. That's where it first was found, and it has been moved to a yeast. All right, so now if we look at the yeast sequences themselves, right, and we ask how are these organisms, how are these yeast related to other yeast? So you have to look at yeast sequences now and compare them to other yeast. So they're not using the bacterial sequences. And they compare them, what they get is, okay, so now you have Wickerameae. I still cannot deal with this marker here. Okay. All of this group, and by the way, the length of these lines that are connecting, this is called a phylogenetic tree, the length of these lines that connect all these members just represent about the amount of genetic distance there is, the genetic difference. You can think of it as if you just lined up my sequence and your sequence for 1,000,000 base pairs, we'd find out that you and I aren't 100% identical. We would have several base pair differences. And so you and I would be connected, say, like very closely together because we don't have many differences. But maybe you and I compared to somebody else that's more distantly related to the two of us, we would have more differences between us. And so therefore, maybe we would be together as part of this group and so forth. Well, all of these that have this name Wickermiella, that indicates they're all part of the same genus, but they are different species. But nonetheless, they're all considered to be sort of similar to one another. And you can see that they form not exactly a group, but they all are similar to one another. And then there is another genus called Starmerella. I get used to saying that, Starmerella. And they are all contained in one group. And by group, I mean there's what you could call a common ancestor. It suggests that there was one species back here that then over time, through mutations, has resulted in a bunch of different species of this. And all the red ones here have the ability. They have the ENT genes, right? All these pluses, right? They actually have in their genomes all the genes from those bacteria. And then if we go back here, these are other yeast over here. These are other yeast, and all these yeast, they do not have them. And if we compare it to E. coli does. Now there's a dotted line here. I have to point that out because you might be looking at this going like, oh, E. coli isn't that much different than yeast. The reason for the dotted line is because it's saying there's so much difference there. They are so radically different. We're not really connecting, and we're just saying like over here, if you go outside the yeast, which is a eukaryote, and you compare it to a prokaryote, E. coli, that's how far away you'd have to go to find another organism that has the ENT gene, right? You can't go to animals, you can't go to plants. You'd have to go all the way out to this bacteria to find another one that has it. So what they're proposing is that somewhere around here, if you go back in time and you were to look at all these species of yeast, and you were to work backwards, just like you would in like a human family tree, right? You and I share a common ancestor, and you and I and everyone on this earth share a common ancestor. So if we're all part of the same Homo genus, right, we would track ourselves back, right, to some common ancestor. You can do the same thing with these fungi. You can say like, if these yeast all, in fact, these two genera, all trace their history to here, if we were to say that this bacterial version of Enterobacter, the genes got moved over somehow into this ancestor, that ancestor then would have shared with its descendants those genes. And so here they share them, they go down here and they share them with all these descendants. The ones in black don't have the genes. Now, how would we explain that? There's a simple explanation for that, and that is they've lost them, right? It's not that they necessarily didn't ever have them before, but maybe in their ancestor had it, but now they've lost that set of genes. Maybe they live, actually, it's not a maybe. The ones that they've looked at that are in black and don't have those genes, those particular species live in much higher iron environments, and therefore they don't need to make enterobactin. And you know what they say, if you don't use it, you're going to lose it, right? And that's what happens in genetics, right? An organism lives in an environment where they don't need a set of genes for a long period of time. Anytime there's a mutation or something happens such that the gene is lost, the organism is kind of like, I don't really care about that loss because I wasn't using those genes anyway. And so you can have, we'll call it decay of the genome at that point, right? It's just decayed it away. But any species that can find survival value in maintaining those genes will keep them. And so all the ones in red, you could say, they're the descendants that have decided to keep that particular set of genes. Now, what's interesting about the descendants is all the descendants have been subject to a bunch of mutations. If mutations occur that get rid of those genes and they're in an environment where they didn't need them, we just covered that, they could just lose those genes altogether. But if they have mutations to those genes in an environment where they need to collect iron, they could select some beneficial mutations or new arrangements of those genes that make their system even more efficient and better at procuring iron. And so what we see is that some of these in Wickerhamelia, they've kind of kept the original set of genes in basically the same order that they're found in bacteria. They've all lost a set of genes from the bacteria, but they have this gene and they have this group of genes and they're in the same order. And Wickerhamelia, they've kept those genes in pretty much that same order. But in the ancestor to the other genus, so as these species changed over time, and then one species changed enough that we'd start calling it a different genus, Starmerella, they had another, we'll call it an evolutionary event, right? They had the insertion of another iron gene, this so-called FRE and TM. So these are two different genes right here. They had an insertion inside or between what were two genes of the bacterial system. But these aren't bacterial genes. These are actually fungal genes. And you can take those sequences out, compare them to other fungi. And yes, they're fungal genes. In fact, they're fungal genes from somewhere else in the fungi genome. And so I believe, I didn't look into this, but I believe they're probably duplications. So somewhere there's another copy of those. But you can think of it this way. If that gene has a particular function that has to do with another iron collecting system, remember that first graphic where you saw that there was multiple ways that fungi can obtain iron from their environment? And now you copy one of those genes, which this gene is involved in one of those iron securing mechanisms. If you copy one of those genes and insert it into this enterobactin system or siderophore biosynthesis pathway, you could add a new feature or quirk to that particular biosynthetic pathway, allowing you maybe to be more efficient at making that enterobactin, allowing you to gain more iron from the environment. And so the duplication of that gene, I would say, adds a new feature. All right, it's adding more information to the genome because now you have two copies of it and each one is doing something different, right? One of them is doing its original function somewhere else in the genome as part of another biosynthetic pathway. But over here, it's being added to this new novel biosynthetic pathway, changing it so that it performs its functions a little bit differently than it did before. So it's now a little bit different than the bacterial version, because it's working with the bacterial genes, doing something different. And then look down here. we've got this other genome that was sequenced. They haven't sequenced all these genomes, so they don't know the gene structure of all these organisms. They just selected a few, and every one they selected was a little bit different from each other. They looked at this Starmarella apicola, and that one, they discovered the genes have been broken into two separate sets. Here's a set here, and here's a set here. I haven't told you yet that these genes are still acting like operons, even though they're in a eukaryote. So this is a really interesting melding of two different regulatory systems from two very different types of organisms to sort of do something different than it does in either one. Really cool stuff from a genetics perspective. Um, there was an addition, all right? So what they're saying, translocation, a movement of genes. In this case, I think it's a movement so that it doesn't exist somewhere else in the genome, and now they just moved it into the bacterial operon and plunked it in right here. And then so it still has these other genes, which it got from its ancestor back here. But then it went ahead and this particular, or these two, probably both of these species, have added new features to this genetic set or this genetic system down here, giving it even additional properties that it didn't have before. You see how what we're doing is we're showing how after an initial event in which the organism procures a whole new set of genes that it didn't have before, it's now playing with those genes. It's saying, hey, let's add in this other gene, right? And maybe we can have a new function that way. And let's break up the genes into two sets. And now maybe each of those sets can do slightly different things, right? Giving us new capacities. The other thing that's happening here, which is less intuitive, is that the genes are getting farther and farther apart. Now, remember what I said about the E. coli genome? I said that the genome was the circle and it's really compact and all the genes are right next to each other and there's really no space between them. That's different than your genes. Your genes are highly separated with lots of space in between. And when the spaces in between give you places to do things like regulate your genes in many different fashions. So we add regulatory elements between the genes because every one of our genes is separately regulated. And so what's happening here is over time, some species of yeast have added pieces of DNA. I'm trying to find one that's a good example here. It's hard to see, but they've added maybe hundreds of base pairs in between the two genes. And what has happened in that space is there have been mutations that have created new promoter positions, allowing some of the genes to be regulated separately rather than all as an operon. So they're sometimes acting as an operon like a prokaryote, but then sometimes they're regulating the genes separately, allowing them to only use one gene and not the others, which is more sophisticated. And so you see in this group of fungi, yeast, we see multiple different levels of sophistication of this particular ceritophore biosynthetic pathway. And it's almost as if you're watching these organisms adapt and change their genomes, procuring new functions, new pathways, new capacities, right? This is all the way that organisms gain information and gain new functions and adapt to their particular environments, all seen at the genomic level. All right, so, this is just to emphasize that this is a picture of one of these yeasts up here, and this is a picture of the yeast down here. They're budding yeast, so they grow in long chains. These yeast over here are not all that different from the phenotypes of these yeast here. However, they're in different groups they're in different genera. Genetically, they're a little bit different. That's the length of this line here. And so the properties of these fungi, even though they look very similar, they can grow in environments that are fairly different than the environment that this one lives in. So in a way, you could say that the addition of this new genetic information right here has allowed these bacteria to invade or better live in more difficult environments that their relatives, because these clearly are close relatives of these yeast right here, these yeast can't live in those same environments. And so that is the addition of a new function that makes these fungi significantly different, right, functionally than those other ones. All right, we have a lot more data yet to look at. All right, we're still building the picture up here to some extent. So I'm trying to remember if I'm forgetting anything before I move on. I'll come back if we do. Ah, now we come to the question of how? You know, how did this original exchange of genetic information occur? In order for you to have an opportunity for a bacteria and a fungus to share DNA, for one to be transferred from one to the other, you have to look for places in the world where the two organisms may live very close together. Where might there be a type of bacteria that has enterobactin living in an iron poor environment, would also be living with a fungus that's living in that same environment that's also an iron poor environment, right? Where would you find that? Well, there is a place where you can find that. And it's a very common place, and that's in the gut of insects, all right? In the gut of many, many insects, there live bacteria that are E. coli related bacteria, kind of like your guts, right? And they provide a number of different functions for the bacteria, the insect, including helping with digestion. But they also contain genes that often make products that they overproduce and that they then give or excrete, and then the host can pick them up, the host insect, so it helps them. So this is a mutualistic relationship because the insect also is giving a home to them. Often they're so related, so dependent on each other that the insects will actually share the bacteria from their gut with the eggs that they lay so that they're sure the next generation will also have the same bacteria. Well, there's also fungi that live, yeast, that live in the guts of many insects. Those yeast are also living in that same environment, and this environment is a very iron poor environment. Iron's a very important element for the survival of cells. So the bacteria living in the gut and the fungi living in the gut have to be concerned about getting iron. So there you have a situation where you have a bacteria, you have millions of bacteria, all with a genome that includes this set of genes for making enterobactin. Some of those bacteria are dying, right? And their cells are splitting open. Their DNA is basically laying out inside the gut. And 99.999999999% of the time, that DNA is going to be destroyed and reabsorbed by other cells and used in the form of energy. But once in a while, and yeast have this capacity, right? Any molecular biologist will know that yeast have the capacity, along with other bacteria, to absorb DNA directly into their cells from their environment. And so when the bacteria dies, its genome is broken into little pieces. If a 16,000 base pair piece that happens to be the operon, including the promoter region up front, the TATA or the TATAAT box, the Pribno box, if that segment of DNA, which just by chance in the, I'll say, quintillions of times that bacteria have lived and died inside the insect of a gut of an insect, in the quintillions of times that has happened, if one time the fragment that consists of just that portion of the genome gets absorbed by a fungus, there are mechanisms by which DNA absorbed into a cell like a fungus can insert that DNA into its genome. Right? Now, I know it sounds pretty wild, but we can show this in real time, really happening. And this is what molecular biologists do all the time. We take advantage of this because we do transformation. We actually have fungi and bacteria that absorb stuff from the environment. We're intentionally giving them genes and having them absorb them. And once in a while, the fungus will add it to its genome. Now, when it adds it to its genome, that's still going to be a random event. It's not going to know where to add it. It's going to add it to some place in the genome. Maybe this happens thousands of times, but never gets added into a place where it can be useful. But if it adds into the right portion of the genome, now think about, I already told you about eukaryotes, they have lots of space between their genes. And in that space, some of it is not all that useful. And so if you insert something into that space, it doesn't necessarily hurt the host organism at all. They're like, okay, I got 3 billion base pairs. what's another 16,000? He's also like, what's another 16,000 base pairs? That's not going to hurt me. And so it sits there. And most of the time, that sequence will never get used, right? If you search the sequence of a fungus, if you search your own genome, if you were to sequence your own genome and then search your genome for foreign pieces of DNA, you would find little fragments of viruses, of bacteria, and even other types of animals inside of your own genome. Because through history, not necessarily your own history, but your ancestors' history, your ancestor might have been infected by a virus that inserted DNA into, like the HIV virus, right? The HIV virus actually inserts its own genome into the host genome and becomes part of their DNA. So it's a known mechanism, and it does happen. Most of the time, those insertions don't mean anything, just like all the probably millions of little bits that are inside of you that are not actually human DNA, but DNA from other sources. Most of those little bits are absolutely useless to you, right? They have no function at all. And you just sort of carry them along because they don't really hurt you, right? They're kind of neutral. But this particular sequence inserted into the fungus, right, seems to have had the whole operon structure. So actually, not the regulatory gene, but this whole segment right here seems to have been added into a fungal genome, into a space that didn't hurt it. And because it had a promoter and operator, then the question becomes, how did the fungus learn how to use these genes? If these genes are foreign genes using a foreign non-eukaryotic regulatory mechanism, operators and promoters, what happened there? So let's go back to our promoters. Now, remember I said you have to go just 10 base pairs upstream to find the Pribno box, which is TAT-AAT, and then that will help stimulate the beginning of transcription and then eventually translation. And I said, but it's 25 base pairs long, 25 base pairs upstream in a eukaryote, and it's TAT-AAA. All right, now here's where it gets interesting. I think it's been interesting all along, but here's where it gets even more interesting. If you were to take the eukaryotic version of the protein that binds right here as the promoter, and then you were to take TAT-AAT, this protein will bind to this, just not very well. All right, it binds to it a little bit. I told you earlier, proteins and enzymes are a little bit sloppy. They have a specific sequence that they're the best at binding to, but they can bind a little, much less efficiently to other sequences. In this case, these sequences aren't that different, and they can bind to it. Now, when this was inserted into the fungus, it would have only been 10 base pairs away. Only 10 base pairs away is sort of like cramped in terms of like how the eukaryotic wants to function. This protein is really big. The proteins that want to bind here to start the transcription process are big and clunky. And so this isn't going to be very efficient, but it doesn't have to be efficient. It doesn't have to be efficient. All it has to do is work a little bit. And it can be shown that it does work a little bit, right? It's less efficient. So you could take this bacterial thing, put it into a eukaryote. The eukaryotic regulatory system looks at it and goes, yeah, that's not really all that great. However, I do kind of see the same type of signal here, and I can kind of hook onto it. And so instead of being, okay, let's give an efficiency number. Let's say that in a eukaryote, if you had the right sequences at just the right places, and you wanted to turn that on, maybe you could do, you could make, 100 transcripts, over the period of a minute or two. But this particular one, because things are in the wrong place, it's not quite the right sequence. It's not, all the conformations of the molecules aren't quite right. It's going to be less efficient. So let's say you only make 2 transcripts per every couple minutes instead of 100, right? You're only 2/100ths of the efficiency. But if you make the transcript, what will happen is And what you're doing now is you're making the transcript of multiple genes at one time. The eukaryotic translation system, which is the machine that reads that code and turns it into proteins, it's perfectly capable of doing that. And so it'll read the enzymes. Once it reads the enzymes, that cell is going to be able to make enterobactin, right? Because all the enzymes are going to be there now to do the metabolic pathway. It makes the enterobactin. Now, what if there's a fungus living inside of inside of an insect, and it all of a sudden it's got these genes and it can turn them on even though it's inefficient, and it makes a little bit of enterobactin, right? So it makes a little enterobactin, and then because it's making enterobactin, it's getting more iron than it was before. What if it was really starved for iron, which these fungi are? Usually their growth is limited by the amount of iron in some cases. So what if they can get more iron? That particular fungus that has this particular, we'll call it a mutation, right? Because that's what it is. This is a form of mutation. A new chunk of DNA has been accidentally inserted in its genome. That is a large mutation, but it's a mutation. That mutation has instantly given the fungus a better capacity to survive in the environment. It might give it a slight advantage. Now that fungus might be able to grow and divide a little bit faster than its neighbors, and as a result, it becomes more common, in which case that particular mutation will be copied over and over and over and over again and become common within that particular species. It might completely replace it in that insect. That insect then spreads it to its offspring, which then begin to make more copies of it and so forth. You see what's happening here, right? Natural selection is taking place. But here's the other important thing. And this is what they see has happened to these sequences. TATT, sorry, TATAAT, how different is that from TATAAA? Not different. What if after the insertion, 10 generations, 100 generations, 1,000 generations later, which may only be a year or two, in terms of fungal growth, what if one of those fungi has a mutation and the T, erase ink, what if this T has a mutation, changes it to an A? That's a mutation. That's a single nucleotide polymorphism. That T changing to an A, you can predict it, right? It's going to increase the efficiency of making enterobactin because now it fits the regulatory protein perfectly, just like a eukaryote. In other words, we just turned the prokaryotic sequence into a eukaryotic sequence, regulatory sequence. Now the efficiency goes up. You start making more enterobactin. The fungi is even better off, right? It's more fit for its environment, and it will begin to do better than its neighbors. Then what happens if you have a slippage as you're copying DNA and you add 5 more base pairs right here, right? That could happen. You have a little repetitive sequence, TATAG, TATAG. You end up with 2 copies of that instead of 1. That's a common mutation that occurs. And you might say, that's a neutral mutation. That doesn't, it's not neutral if it happens here, because what it does is it spreads out the distance between the gene itself and the Tata box. That makes it more efficient, right? Because before it was 10, it was still cramped. Now it's a little bit farther apart. It's getting closer to that 25. The more mutations, each mutation that adds more sequence there, and you can say, that's brand new sequence, by the way, when you add that sequence. I know creationists will say, well, that's just a duplication of sequence that was already there, so that's not new information. I was like, it's actually new information, because if you add more sequence, it doesn't even matter what the sequence is. If you add more sequence and you push the 2 sequences farther apart, you create greater efficiency. That greater efficiency is adding a new feature to the organism that didn't have before. It now can make a lot more enterobactin than it could before, the day before that mutation occurred. And so that new addition of base pairs makes a difference. And that's going to be selected for. Now, do you see how if you just keep on having more mutations over time, you're gradually going to adapt a system that was only inefficiently working as a prokaryote system inside of eukaryote, and it's eventually going to transform it into a eukaryotic regulated system. Now you can ask the question, do we actually see that in our sequences? Now, yes, we do. And what's really interesting about all the genomes that they sequenced is they're seeing that the sequences, I mentioned this before, they're farther apart, which means they've had mutations that have added sequence between. And you know what happens if you add 100 base pairs between two genes? There's going to be some sequence that has T, A, T, 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 A, T, A, A, T, A, A, T. There's going to be combinations of T's and A's. They may originally, in the original version of like what was the accident that added more DNA, they may originally not be the exact TATAAA Tata box. But once they're there and it's TATAAT, like a prokaryote, now you have an inefficient promoter box, promoter site. And so all of a sudden it's like, oh, wait a second, I can actually tell this gene to copy starting right here going forward. I don't have to start over here every single time. And then if that has another mutation and turns into a Tata box, you get even more efficient. And then other genes have additional additions of genes that have their own promoters in front of them because they copied them from somewhere else in the genome. So we're remixing these genes and then we're adding more space. And then when there's more space, there's raw sequence to work with that you can then have mutations, which will then create new promoter sites, new DNA binding sites. The new DNA binding sites then give you new opportunities to regulate the sequence. And the more you can regulate, the more different things you can do. You can be like, I don't need enterobactin right now, so I'll just turn it down. That's regulation. Oh, I need more enterobactin because I'm in a much more poor, iron poor environment. I can turn it up by simply making more of these DNA binding proteins, which will bind to these new sites, which then increase the amount of enterobactin that's made. It's a really cool system. And what I'm trying to also communicate to you is how dynamic molecular biology systems are, all right? Biochemical pathway systems are. They're very dynamic. And they're not as static as you might be led to believe. Like if you just change one thing, you destroy everything. No, it's usually a question of gradation. Like you make something less efficient or more efficient, but it's not always completely knocking something out. And so that's why organisms can adapt and change. Is because they're not simply on and off. They are mixtures of things. Okay, so I promised the next step is you have to show that not only are the genes present in these species. So here's Starmorella bombicola, Starmorella apicola, two different species, very similar fungi, but two different species. I mean, so I think young Earth creationists would certainly say those two species must be the same kind, right? God must have made that same kind, and then somehow they diversified from each other. How much do they diversify? Well, this one has a different gene inside of this genetic system, this operon. And these two, they have the same genes, but they've also split the operon into two locations. There's actually a large gap of DNA right here that I don't know what's in there. There might be other genes in there, but they're not related to the system. So that shows you that even their genomes are diverging from one another and they're learning to do their own things. Once the species split to become two different species, they now no longer are sharing information. So therefore, any new mutations they have make them more unique from each other. Now, the hard thing to explain is this graphic right here, C. Number of reads. What they're trying to do is they're trying to show, and this is, it's this one here. This is the other genus, and here is the gene map of it. Here's all the genes from the bacteria. And they're saying how much transcript is made from this gene. And the height of this is saying how many copies of the reads are existing in the cells. There's a way you can go in, you can extract the messenger RNA, the messages from a cell. And if you extract the messages that are inside of a cell, inside of a yeast cell, that'll tell you how many copies of a gene are actually being made inside the cell. So that's your first step to showing hey, these organisms are actually using the genes. They're not just in the genome. You can show that by just sequencing the genome. But I could sequence my genome and you could sequence your genome and you could find pieces of genes in your genome that you are actually not using, but they exist there. But we have hundreds of genes that we don't use at all, but they exist in our genome. So in this case, we want to show more than, I mean, the whole purpose of this is to show that an organism can gain genetic information, right? And useful genetic information that they turn into novel features. That was the criteria that Brian Thomas said in his quote, in which he said, this has not ever been shown, right? I'm showing you an organism that has gained an entire piece of genetic sequence and code, and it's reading that code. That's what all these humps are, all the reads. It's reading the code. And then what I'm not going to show you is they did the further experiments to show they make enterobactin, it's in the cells. And not only that, they can put the fungi in different environments with different amounts of iron, and they can show the fungi get the environment from the environment. Sorry, the fungi get the iron from the environment and pull it into their cells and store it there, which is what that enterobactin is supposed to do. So they can show they functionally do it. And so, all right, back to this graphic. These little bars are saying that they do transcribe all the way across 1, 2, 3 genes, all the way across here, all the way across here. This gene is actually read separately. Oh, I'm sorry, this graphic is, I forget, this graphic is related to this one up here. Anyway, this particular one, all of these are read together as one long transcript and then translated in separate parts. That's an operon, right? The fungi don't have other things like that. They do all their genes separately like you and I do. But this particular fungus is accommodating the prokaryotic system by reading them all together. But they also don't read them all together because there are some that are read more often than others. And that's because there are some of these, as I was saying before, some of these in the space between them, they have their own promoters that have evolved there from de novo, from just raw sequence, random sequence, they have turned into promoters. And that way they can turn on some of the genes separately so they don't make the same amount of every one of these genes. And that is how they are beginning to act more like eukaryotes. And another part that's compelling about this whole story of how this whole system is evolving is some of the fungi are more prokaryotic like, right? They haven't had the random mutations to allow them to get the promoters in the right spot. And other species have, I guess you could call them, they're the lucky ones. They've had particular mutations that have inserted just the right TAT-AAT or TAT-AAA code in the right spot to allow suddenly them to be able to regulate those genes separately. And now that they can regulate them separately, they have more options in terms of how they're going to use those genes for biosynthesis in their particular environment. So each of the species is becoming more, in this case, they're becoming more sophisticated. They're getting more complex, if I could use that word. Clearly, these fungi are more complex than their relatives, which don't have this system. And they're actually increased their own complexity even after they got the basic system from the prokaryote, from the bacteria. They have added new things to it, allowing it to do even more than the bacteria could have done with it before. Yeah, just an amazing system. So what have we observed? We've observed that some species of fungi in one particular group of fungi, one particular group of yeast, have the capacity to make the molecule enterobactin. This molecule allows the fungus, which all grows in a low iron environment, to make more efficient, to more efficiently scavenge that iron from its environment. So it has a competitive advantage, right? That's a novel function that gives it an advantage over other organisms in the same environment that can't do that. The genes then enable this distinct physiological ability or phenotype are very similar and in the same gene order as that found in bacteria related to E. coli, right? The genes themselves that are making these enzymes are still making the same enzymes that we see made inside of E. coli and other bacteria that make enterobacter. So really, they haven't adapted those, right? Because they do the job well. Like the protein itself, the original code from the bacteria is still really good code, and they're still using that. They've just adapted the regulatory elements around it to make them be more efficient at making those particular proteins. So closely related species of fungi with this capacity have different genomic characteristics, including the addition of other fungal genes into the operon, providing additional functions and phenotypes. So those, even after you had the initial addition of genes from bacteria, the fungi have added other additional genes to that. Now, when I say added, I don't necessarily mean that they have a mechanism where they know what they're doing. It's just that the genome is always having accidents and moving things around anyway. And if one of them happens to move a gene into an iron gene into this iron system, the fungi is going to go, well, thank you very much. I mean, that helps me out, right? It gives me an advantage. And so it'll begin to copy that more. In the same way that a mutation that creates a ta-ta box from a Pribno box is going to increase the functionality for that particular fungus. All right, so those are all the things we've observed. And what I would say in the end is we have observed a fungus which did not have the capacity to make enterobactin and survive as well in its particular environment as it could after it obtained a whole bank of new genes from another organism. And it has sculpted those genes even more to become efficient inside the fungal system. And it gives them a novel trait. They have obtained a novel trait. So what are the objections to this? Like what are the creationists going to say who say that no new novel information can be ever added or gained by an organism? I know what the first thing is going to say. They're going to say, this isn't new information, right? They could say, by I think ignoring the, I'll say, obvious intuitive answers to the data I've shown you. They could say this was just God's original design. All right. He made these particular fungi with these sets of genes and metabolic pathways to make enterobactin separate from any other fungi. He just made those fungi to have this ability to make enterobactin. And maybe God, in designing those fungi, just looked at those bacteria and said, that's a good way to do it. I made for bacteria, and I'm simply going to copy that set of bacterial genes, and I'm just going to put it in the fungi and have the fungi use those, right, as my design. But they didn't actually transfer them there. This wasn't part of history. This is just the original creation, and I'm just using the design, kind of like chimpanzees are really similar to humans. Why is that? It's because God uses the same design for the basic body plan, but then adds different features in order to make them different. Couple different problems with this. One is this isn't the genes that they have now aren't an exact copy of those bacterial genes. Yes, those genes are same, but the fungi have adapted them in different ways. And so they're not clearly well designed originally to work in a fungus. They've had to completely sort of add new features to it. And you could say, well, maybe that's what God did. God just had in his mind the enterobactin system and thought it might be nice if these fungi had the enterobactin system. So I'll create it in them and using this bacterial system, but that's a prokaryote. So I'm going to sprinkle in some features from eukaryotes. It's still kind of a mixture of the two, right? If you were just designing it straight up front, you would say, here's the most efficient way to make enterobactin, and you can do it without a prokaryotic system, but I'll give you the genes in a eukaryotic system to do it. What we see here is it looks like a transition. It looks like the evolution of this system from one prokaryote to a eukaryote. Also, second objection, really, which might be the first objection, might be the most common one. This isn't new information because it's just using information, DNA code, that already existed elsewhere, though in a new context, right? And this is how they explain away things like gene duplications inside of an organism, where you would say, what if a set of genes gets copied, so now you have two copies. Now one of the copies can have mutations that change their function, while the other copy does the original function. So over time, what you can end up with is two different functions based on what was originally one set of sequences. And this has been shown to occur, but creationists will reject that and say that, but that's not really new information. You just copied the same information over again. So the original information was just, God created that original information, and all you're doing is degrading the other copy somehow. Even if it makes a new function, it's degradation because it's not the perfect copy it was there. Okay, sure. Even in this example, this is a copy of information from somewhere else. So I'm not talking about where the original information came from in terms of the actual like DNA sequence of the enzymes themselves, the genes themselves. These aren't new genes in terms of like brand new to the existence of the world, but they absolutely are new genes to the fungus. And Brian Thomas said, he was talking about within a kind. God created this, yeast, and no new information could ever be added after that. Well, here we have an example of DNA that's not from yeast now in yeast, but only in one particular group. It suddenly shows up. It's suddenly there, and it has a novel function. But there's another thing too. The reason I spent so much time talking about some of these other features is new information is anything that adds to the capacity to the phenotype of an organism. So if you change the Pribno box to a Tata box, allowing for these sequences to be more efficiently copied, you change the phenotype of the organism because now it's more capable of sequestering iron. That's a new feature. Better able to than your ancestor. Well, where did that change come from? That was a mutation that changed the code from one thing to another, allowing for a new feature. Now, creationists still object to that. They say, yeah, but you didn't actually create the new code, right? There's still code there, and that code had to change. And even if I said you copied the code into 4 different pieces and then changed one of those pieces of code, they would still say, well, but you copied another piece of code. That's a chicken and the egg argument, right? That's essentially saying because you're not explaining the beginning of the world, therefore I don't have to believe anything else you say, and I can just say, well, you didn't explain the beginning of the universe, and so because you didn't explain the beginning of the universe, it doesn't matter how much you can explain about how things work over the last 1,000 years. I don't have to accept your explanation. And besides, that wasn't the challenge. Brian Thomas had a particular challenge in that quote. This example meets that challenge and says that kinds can obtain new information and new characteristics. And if you take this and I'm just given one example here, right? This is only one example of hundreds of examples, and I'm sure millions of examples that actually exist, but hundreds that have been described. This is what organisms do, right? They are shifting and changing. They're adding components. They're subtracting components. Creationists always go to the subtraction, right? They can give you lots of examples of subtraction and say like, okay, yes, you changed features, but that's you lost information. But in reality, there are many organisms that are adding new information like this, adding new characteristics. And yes, it's changing the nature of these organisms such that they are changing their basic operating system over time and becoming different kinds of organisms as a result. So that goes to my last objection. These fungi are still the same kind of fungi. It didn't change them into new kinds of fungi. Well, this specific change might not have changed them into a new fungi exactly, but I would say that the addition of this certainly made these two genera different than any of the other genera, which is a way of saying they're different, right? Because they're in a different genus now. They're a genus that's characterized by not just siderophore biosynthesis, because not all of them can do it. Some of them have lost this capacity. But being able to live in this environment better has allowed them to live in an environment maybe they couldn't live before, and now they've also adapted to living in these very strange gut flora environments, which has made them different than their neighbors, which aren't living in guts. And I'm sure if we were to search more, we'd find hundreds of other characteristics which are different, probably as a result of them being able to survive in this environment because they can get iron more efficiently. And so now they can do this, and they're working on that, and they've adapted to this, they've adapted to something else, they've adapted to something else, they've adapted to something else. They're adapting to 100 different things. They, in fact, have become different types of yeast, different kinds of yeast, if you will, over time. And this just shows, in principle, new features can be derived in organisms in a variety of different ways. And the accumulation of those variations can certainly derive significant changes in organisms over time. Well, that's my story about adding genetic information to organisms. If you've stuck with me this whole way, then you certainly are you're interested in nerdy science. And I appreciate you coming all the way with me. Hopefully, if you didn't know any of this material before, hopefully you have a greater appreciation for, say, maybe the complexity of genomes and gene regulation. Again, so I didn't even introduce myself at the beginning, but my name's Joel Duff, and I am professor of biology, and I teach genetics, and I tell stories like this in class. Now, stories is a bad word. I go over research like this in, say, an advanced genetics sort of situation. I give examples of, like, the things you're learning about. Here's an example from the literature of what's actually happening. So thanks very much for listening to my monologue, my lecture. And if you found this information useful, go ahead and subscribe to my channel, where I'll probably review some other papers in the near future. Thank you very much. Thanks for listening.